I'm very pleased to have Dr. Joe Jarvis here today. This is for the third Genomics for Teachers session this semester. And Dr. Jarvis is the Director for Genomic Risk Reporting at Coriell Institute for Medical Research. And that's just in Camden, New Jersey, so just across the river. He is running the Coriell Personalized Medicine Collaborative, which is a longitudinal research project aimed at identifying the best practices for the use of genomic information in healthcare. And his background is in quantitative and statistical genetics. So without further ado, Joe.
Thanks so much, Kristen. It's a real privilege to be here today. I do have a reputation for using jargon, so if I say something, word or phrase that doesn't make any sense, jump on me. Feel free. I wish I had one of those taboo buzzers. Feel free to raise your hand, make some noise, let me know if there's something that's not clear. So I'm going to tell you a little bit about Coriell Institute, just very briefly. So it's a nonprofit research institute that was founded in Camden a little over 60 years ago. The founder, Lou Coriell, was actually involved in the original Salk polio vaccine trials and actually invented a lot of the technology that makes cell culture possible. And so the ability to take cells, put them in a dish, grow them up, and actually manipulate them in vitro, in situ, is something that he actually helped to pioneer. And so you can see here we've got tanks of liquid nitrogen containing all sorts of different samples. It really ends up being the most diverse collection of human cells, DNA, and other biospecimens in the world. So a lot of our funding comes from NIH and other sources. One of the things that I always like to highlight when there are academics in the room is that one of our longest running funding agencies at the NIGMS contract has been continuous since 1972. So there's nothing quite like continuous funding, and it's been a privilege to serve the NIH for so many years. So we're going to talk a little bit about genomics and personalized medicine. I'm going to be perhaps a little bit redundant early on with some of the terms and such from both the last session and maybe early on. I hope that's helpful. Again, everyone defines things slightly differently, but I want this conversation to be sort of one that we're all on the same page for. Also, please feel free to raise your hand, any question at any time. I would be fine if this devolved into a discussion rather than me lecturing at you. Giving you the information that's most helpful to you would be just fine with me. I can babble about anything for as long as I need to. Believe me, ask anybody. So we'll start, of course, with cells. So they're incredibly different looking and function completely differently in the same organism. So these are striated muscle cells, and you've got neurons, you've got all sorts of different cell types, all expressing DNA that should be identical. So I will sort of throw in a couple of different things. Somatic mutation does happen. So after you have a fertilized zygote and it grows up, you have a bunch of cellular multiplications. It is possible for mutations to be introduced during the process of development post-fertilization. So unless those happen in cells that are destined to be in the germline, in the gametes, the sperm and egg, those mutations never get passed on to the next generation. But they are mutations that might actually exist, and you can get really interesting situations where we are all mosaics, in a sense. And so it's not only possible for the same DNA to be expressed in different ways and manifest in different cell types, it is also possible for different cells to have slightly different genetic complements. So a lot of genomics is in some ways a throwback. So humans have been doing genetics for a very, very long time. It's just we haven't really always called it that. And there's a lot of legacies that spring up because of this. So agriculture. There's been all sorts of controlled breeding programs where humans have been manipulating the genomes of organisms for tens of thousands of years. Well, I guess 8,000 years for some of the domesticated crops. But we've also seen fortuitous mutations occur in our stocks of both wildlife, domesticated cattle, sheep. The example up there is an Ancon sheep. So what you'll notice is that it has very short, stubby legs. If you are a farmer in New England in the 18th century and you have to build your fences out of stone, this is a very advantageous trade. They can't jump a very high fence. And so really these have occurred multiple times throughout history. There are European breeds that go back further. Very often they end up getting bred away, they go extinct, they pop back up again. So these mutations actually have been arisen and manipulated by humans for our own purposes for actually a really long time. I also like to highlight the white-eyed fly here before I get into the Mendelian stuff below. So Thomas Hunt Morgan, same sort of thing, decided to use, instead of agricultural organisms, used fruit flies They're convenient. I'm sure many of you are familiar with the use of fruit flies and some of these mutations. He happened to just plain observe a single white-eyed male, then bred it out and observed some of these patterns and discovered the sex-linked trait. So it is actually linked to the sex-determining chromosome, which gave a very interesting pattern of inheritance, and it sort of played into a lot of his work eventually. He ended up winning the Nobel Prize in, I think, 1933 for contributions related to the chromosome being the structure, the physical structure that bears genetic information. So it's actually fairly recent, right, that we've been doing genetics with genotypes. What I've been describing now are genetics with phenotypes, traits that you can observe, that themselves are markers for the underlying genetics. And so now as we've moved into being able to directly observe the DNA, our markers are a very different type. They're a molecular marker. But we've been doing genetics by tracking traits for a long time. What I would point out that is the difference, and we'll get to a lot of these different nuanced layers, is that the genetic architecture of these traits are very different. By architecture, I mean the number of genetic loci, places in the genome that are involved in variation, the ways in which they combine their effects, the size of their effects. What you're looking at here in all of these examples, including the Ps that we'll get to just momentarily, are very large effect mutations at very few number of loci, single loci. And so these are the textbook examples of Mendelian, so-called Mendelian traits. And so a lot of genetics, that was what we had to work with, traits that we could observe that bred true. So parents and offspring share characteristics like the short stubby legs of the sheep, and we use those to track. Those were the early examples. And so it's interesting to see the legacy of the Mendelian characters bleeding over into something like personalized medicine, where there are clearly clinical cases, obviously, of high penetrance mutations that happen at one place that have very distinct patterns throughout families. But a lot of what we care about, including heart disease, diabetes, so perhaps even risk for cancer, these are more what we call complex traits that involve perhaps many more loci, smaller effect sizes, with interactions both with each other and with the environment. So the bottom example there is, of course, the famous publication, well, an example, a cartoon of the famous publication from, I think it was 1866, that Gregor Mendel published this work. He was very lucky in his choice of characteristics. Some would say preternaturally lucky. That is, he probably cheated. He probably went out and measured a whole bunch of stuff, found a subset of traits that matched the same pattern, and used those to build his laws. And in the end, he was right. So it really doesn't matter too much whether he cheated. But it is kind of amazing that he picked all loci that were on different chromosomes, that segregated independently, that have very high effects, that were fully penetrant. That is, the presence of the mutation gave rise to the physical characteristic almost every time, or every time. So he was pretty lucky. But so you can see what the kinds of traits he was working with, where you've got the color of the pod, the color of the seed, whether they were round or wrinkled. These are all classic examples that perhaps you've encountered before. But these are also examples of, as we say, Mendelian characteristics. And so these are very simple genetic architectures. And so there are some, as I said, clinical features that follow the same pattern, and many of them don't. And to me, the ones that don't are the interesting ones. Those are the ones that are not only perhaps the larger burden on the medical system, but also just intellectually interesting. Real brief anecdote. So if you're familiar with balancing chromosomes in Drosophila, it's another sort of phenotypic characteristic that stands for an entire chromosome. So there's a manipulation that gets done, there's lots of inversions, and it balls up so that recombination doesn't happen. And so that ball of DNA now, instead of the linear molecule, that ball carries that phenotypic characteristic. And so this mutation here, curly, is one of those. I did a little bit of Drosophila genetics as an undergrad, and I was terrible at it. I was absolutely horrible at it, because these are classic examples, right? And they always show the pretty pictures. In actual work, the notched mutation, for example, which is a piece that's missing, it's not always obvious that there's a piece missing. You have to be really careful. They don't all look the same. Curly sometimes is very curled in others. And so my stocks ended up being terrible because I misread some of these phenotypes. And what that did was really drive me to look at characteristics. Why exactly would that be? I was taught Mendel's peas. I was taught white-eyed fruit flies. Why is there this variable expression? And so my thesis work was on epistasis, genetic interactions, and its consequences. Also maternal effects, which are really crazy interactions between offspring and maternal genomes, and also gene by environment interactions. So that's a little bit about my background as well. Any questions for me so far? Kind of review stuff. Okay, so this is an example of a clinical pedigree, right? So this is the Mendelian kind of characteristic that one might see in a medical textbook, where this happens to be an X-linked syndrome. Some examples that I just pulled are Rett syndrome, Fragile X might look like this. But this is an X-linked and dominant feature. That is, you only need one copy of it to manifest the trait. Recessive, then, when you have two, is the only time you see it. And so there's an interesting, very hallmark characteristics here where, because it's X-linked, it starts with a father. And then all of his sons, all of the squares here on this line, can't possibly be carriers. It can't possibly be passed because it has to have two X's, right? Mm-hmm. So it's his Y and the maternal unaffected X, right, creating the sons. Right. It's only when you get a female where it's his X that carries the disease-causing mutation, and then the other that you end up seeing it. And then you see this pattern get a little bit more complicated in the next generation, and again in the next generation thereafter. So tracing these phenotypes through time in pedigrees, very much like you would have a pedigreed agricultural population, is how a lot of clinical genetics has classically been done. So again, very briefly, what you're looking at here are karyotypes. So these are metaphase chromosomes that have all shrunk back up. They're very tightly packed. I also like to point out here that there's another marker that's been classically used in human and other genetics for decades now that you're looking at right now, and it's the banding patterns. So the actual color, the dark and light colors are repeatable bands. And so at the last session talked a little bit about deletion syndromes, where you're missing an arm of the chromosome. You can see that actually from the bands. And what's amazing is they actually have pretty good resolution. You can, counting bands, especially in Drosophila, get down to a very, very tiny physical region of the genome by again, using as your marker, not a trait, not the molecular part, but the banding situation on the chromosome. I suppose you can call that a trait too, but it's a trait of the physical DNA itself and not necessarily a trait of the organism. There are also, as I would point out, you know the standard 22 autosomes and the sex chromosomes. So this is a karyotype for a male, karyotype for a female, and there's all sorts of new technologies that have gone beyond this simple banding to all kinds of crazy stuff you can do. Chromosome painting and high-resolution stuff that's targeted at very short runs of nucleotides. You can get pretty specific with some of this stuff. And so, same principle, new technologies. And that's really the point of all of this. It's the new technologies that have opened up genomics, what I would call genomics out of the root of genetics, where you're talking really about simple genetic architectures and individual locations in the genome. The difference really is scale. We really can observe much more of the genome in a much finer grain detail. We're seeing things we might not otherwise have seen. But as with every innovation in technology, there are limitations. And so it's very often that you'll only hear the great things about genomics. We're going to talk today a little bit about some of the limitations that are inherent in the technologies. So hopefully that will also be helpful and maybe spur some discussion about what one can actually conclude from the data that are collected. It's really important that your interpretation matches the raw material that you're bringing in, because if you haven't measured it right, you're not going to get a good answer. So obviously, when it spins out, it's this double helix structure, and I put this in just so for reference. When I'm talking about strand, I'm talking one of the rails of this double-stranded helix structure. It's like an extension ladder that got wrapped around a telephone pole, and so that's the piece that you get from mom, and the piece that you get from dad. So there are two of these at every locus for sex chromosomes, but every autosomal locus for mom and dad. And what's often not clear is we use the terminology base pair. This is the pair, right? But you have two pair, a pair from mom and a pair from dad. And sometimes that's not always clear. And so the strand that you choose to report from, the one going up or the one going down, you could either call, for example, at this locus here, you could either call this an A or a T, and you would be correct either way. And so there are some tricky situations that come into play, and one has to make some agreement with the wider community about which strand one's reporting from. Because you can have in a publication one reporting from one strand and one reporting from the other, and if it's not clear, it gets very difficult to interpret some of the published literature. So there are all sorts of conventions inherent in here that can lead to confusion. There is also, as we'll talk about with the reference genome, and we'll get to this, but there are insertions and deletions all the time. So it's not as if you can really start from the left and go to the right and just line them up, because I might be missing thousands and thousands of base pairs. So where do you start? Where's number 1? And what happens if I'm missing 200 to 300? Do I just skip that and start with 301? So we all have to agree on these common conventions. And so for me, one of the most important features of the reference genome that I don't think people really appreciate is that it gave us that common language to talk about physical locations. Previously, it was the P arm and the whatever band. Before that, it was the white locus. They actually called it by the name that the phenotype looked like. But this gives us a whole new nomenclature and a whole new way to standardize work across labs and across technologies. So we hear a lot about next generation sequencing. And we'll talk a little bit about what that is and how we get to it, and there are some limitations there too. But there are also array technologies and other ways, but they all sort of now agree because of the Human Genome Project and the Human Reference Sequence. So we don't need to go through all of these. Does anyone have any questions about them? The only one that I will point out here is haplotype. So there is a difference, and this is one of my own soapboxes, and everyone who sees this is going to be making fun of me, but it is something that I think is really important. The strand that you got from mom and the strand that you got from dad in the abstract are the things that code for the enzymes or whatever functional units, right? So if you have multiple mutations, it's really important to know if they all came from one place or they're spread between both places. With some technologies, and I'll show you why, it's not at all clear. And the next revolution in genomics, in my opinion, again, is going to be true phased haplotype data. That is actually being able to read the chromosomes left to right, top to bottom, beginning to end. My argument is we have never done that yet. There are some exceptions, but we pretty much have never done that before, read a whole human genome. And we'll talk a little bit more about why I make that claim here momentarily. So other than haplotype, which I'll also explain here in a little bit more detail, any other questions? Great, don't forget to buzz me. Okay, so what you're seeing here is a graphical depiction of the human reference sequence. The black dots are the centromeres, so that's the narrowing neck on the chromosomes that you saw in the karyotypes earlier. I've ordered them sort of chromosomes 1 through 22 with X and Y at the top. From this you can see why they're numbered 1, 2, 3, 4, 5. It's descending order in length. You can also see that we didn't quite get it right. 19 and 20 and 21 and 22. Actually, there's one of those that this doesn't show right. I forget which one it is now. I think it might be 19 actually really is shorter than 20, and it should have been flipped. But we maintain the convention nonetheless. What you'll notice here is this is a haploid genome. This is, for example, just the piece you got from mom, or just the piece you got from dad. It's half the genome. So when we talk about the reference sequence having 3 billion base pairs, that means really, in that definition, we all have 2 genomes. We've got a genome from mom and a genome from dad for a total of 6 billion base pairs. One can also, as we talked about with somatic mutations and some of these other features, every cell theoretically has its own 2 sets of genomes. So the genome is a very, very fraught phrase. It's bandied about much too blithely. It's something that we really should probably be more careful about how we say it. But for better or for worse, scientific terms have a tendency to devolve, and that's one of them.
I have 2 questions.
Sure.
Okay, so a centromere, I mean, I'm teaching biology, I have a very, I don't even know that I define a centromere other than a place of attachment.
Yep.
As that. I mean, what is it really and actually? Is it just a placement so that? It's
a very obvious feature on the chromosome from the sequence standpoint. It has its own set of features that don't look at all like genes. So there are enrichments of bunches of A's, bunches of G's.
Oh,
there are actual from the sequence And now I have to remember if I'm actually saying that right. So the heterochromatic regions, both at the ends and at the centromere, actually really are different in their base pair composition.
Okay.
If I gave you a piece of that and I gave you a piece of something else, it would be obvious that it has a higher percentage of certain base pairs.
Okay, and my other question then is, so when the human genome was mapped, you're really saying that sort of like a blueprint of a building these are the structures, and then, you know, I always explain genes and alleles to students, and I am teaching students that are alternative ed, so biology is definitely not their favorite subject. So I talk about genes as in like dungaree genes, and I say alleles are like varieties of those fair. Like, you know, Wrangler versus True Religion versus, you know, that's kind of how I explain that.
So you can also go with flavors.
So when we get back to the genome, you're kind of saying that the human genome, like rough sketch of whatever, and then all these haploid types are all these, like, we couldn't have possibly sequenced every single solitary genome that's even out there.
You got
Just a rough sketch. Okay.
You got it. Okay. Absolutely, you got it. One of the examples that I like to throw out at the HLA region, in these 20 different loci that are of particular importance, if you actually take the variation found at these 20 loci, there are more possible pairwise combinations than there are electrons hypothesized in the universe.
Oh my gosh.
So the number of potential combinations is absolutely vast. The number of actual combinations that have occurred in human history is a very, very tiny subset. So the diversity present in our collective genomes is astonishing. The other thing that's astonishing that I'll also make an argument about is just how same we are, just how similar we are. The vast majority of this stuff looks the same no matter who you're looking at.
Yeah. Thank you.
Answer your question?
Yeah.
Okay. So when you zoom in on a particular locus, and I've chosen one, and I'm sorry might be a little bit small, but this is CYP2C19. It's a locus that's involved actually in phase 1 detoxification of anything that you take in. So foods, herbs, vitamins, actually not vitamins. So, but particularly now, what we're interested in is the pharmacogenomic aspects. So when you take a pharmaceutical, your body processes it just like you process food. And so there's certain flavors of these alleles that do better or worse at their predicted jobs with respect to these particular pharmaceuticals. So this happens to be one locus. But the other thing you can see here is that there are some areas where there are no genes. This gray area is something that doesn't look like what we classically call a gene. There are lots of overlapping. You might not be able to tell, but there are some protein coding regions that might be coded for one strand versus the other. And so the genes reside at physically the same location, but they're read by the body from one strand or the other. So genome structure is actually a fantastically complex subject. And so there was just an article out today. I didn't end up getting to read the finer print, but it was that there was much less DNA in this particular plant species than they expected, and many more genes. And so like something like yeast that has a very compact genome, it doesn't necessarily scale that more DNA equals more information. And there are regions of the genome, quite frankly, we simply do not understand. They were called junk, and they're not. They don't look like what we think of as a functional unit, but clearly they're associated with diseases. And so as we've peered into each other's DNA space, what we found is some pretty fascinating stuff. And we are a long ways from some of the answers that I think everybody would love to have. And that's really exciting for somebody who studies this stuff, because it's sort of job security.
Question, please.
Sure.
Are those areas where introns, exons, are they finding now that those areas are doing something instead of just being a placeholder?
So what I'm showing here is everything black is the protein coding region, including both introns and exons. But yeah, you're right. Within that, you've got the intron and exon components, and they've found examples of pretty much everything you can imagine. So a mutation in an intron that actually does govern expression, perhaps not even of the gene that it's in. Sometimes an upstream or downstream gene, a mutation in a place that's supposed to be just pulled out. It's not useful, right? It doesn't get coded for a protein, except that it regulates the expression of a gene somewhere else.
Are you finding that they are existing in other species in the same place, same location?
There is a tremendous amount of overlap and consistency across species. There are also known trans-specific polymorphisms, and these get really strange, where you can think of, the analogy would be, the allele that I got from my mom is, in terms of the sequence, more closely related to a rhesus macaques than the one that I got from my dad. So these are very, very old mutations in the evolutionary tree that are still polymorphic in these species. So we're finding all kinds of tricky stuff. And there's an awful lot of conservation where species look the same. That's true. Lots of places that they differ too. Does that answer your question?
Yes, I'm just seeing, you know, for so long they just said they were placeholders. They weren't sure their function. And so I was just wondering, has there been any more research on what they actually do with so much dedicated medicine to designing for people?
Sure.
Are mine the same as hers in terms of the spacing?
Yep.
What are they doing there?
There are differences. There are definitely clear differences, and some of them we've shown to have consequences, and some not so much. There are gene conversion events too, where a piece of one gene gets cut and pasted into another, and it inactivates the gene. And so someone who bears the conversion might not have a functional enzyme there. But if we didn't know that, we wouldn't really have expected it. So the reference sequence is publicly available. So these are our tax dollars at work. You can cruise on genome browsers of all different stripes. The UCSC genome browser is one, Ensemble is one. If you put in genome browser into Google, you'll find a bunch of them. And so what I've shown here is actually a flat file of chromosome 21, and what I'm showing is this, this happens to be one of the telomeres, the very ends of the chromosomes, and this is base pair number 1 on chromosome 21. But we know there's stuff to the left of that, and we've put in a bunch of placeholder ends. Partly because our technology can't actually access those areas very well. Like I was saying, they have a different base pair composition, and that makes the chemistry difficult in some of those regions. Repeat regions where you have tandem repeats of the same sequence over and over and over again also tend to trick up our current technologies, where we can't really tell exactly left to right, because that's not how we're reading across the chromosome. So there are some, again, these are limitations that are really interesting and important, and very often are not mentioned.
So how do, I don't want to jump ahead and this diversity that you raise and the unknowing. I mean, talking about medical, personalized medicine and people getting pharmaceutical drugs, even if it's tailored to these things, when it's so much unknown, it's kind of like, I don't want to go down that end when I hear the technical part of it, because I'm thinking of students, how do you get them to understand the critical thinking part and making choices in relationship to this information, and at the same time, it's so much unknown and it's being thrown out like it's known.
Yeah. So that's actually a great question. And so the way that I make sense of this and that I try to teach this is that continuum between Mendelian genetic architectures, the real easy stuff. So if you've got, so imagine an assembly line that's taking a drug and it's turning substrate into substrate into substrate. If there's a rate limiting step in that pathway, if there's one enzyme that's the slowest of the bunch, hitting that one is going to affect the output of the system. And so you can, in many ways, in some cases, the genetic architecture becomes simple because the rest of them are fully functional. It's one that really matters. No matter how complicated the pathway or the cascade, it's one that's the linchpin, one that's the center that really becomes something that's useful. And so those do exist. And what you have is a continuum between the simple genetic architectures like that, where it's one mutation makes enough of a difference that we can see it in a clinical population, and there is a clear clinical interpretation of it, all the way through to the ones where you just throw up your hands and you say, no idea. So there is a continuum there, and what we're sort of doing is pushing the boundaries from that early Mendelian type of architecture, where we're starting to get a handle a little bit better on some of the more complicated ones. But it is still early days, in my opinion. So I don't know if that helps or not, but it's that continuum that...
It's a dialogue, so
absolutely.
Yeah, this, just on something you said earlier, is what part, you're allowed to say you don't want to answer, what part of your mother's genetic material is closer to a rhesus macaque than your father's?
Okay, so all right, so this is one of those...
How did she feel about hearing that?
So this is one of the examples. So I don't actually know my own HLA. I think it's the HLA region that I use most often in that example, and I'd have to brush up on it. But it's a highly polymorphic component of the immune system, right? So it's extreme diversity there because you want to be able to counteract any sort of an infection. And so what happens is there's a tremendous amount of variation there that is preserved in populations. And it turns out when you've got that much, it sticks around for a really, really long time. And it can start to be around for so long that it antedates, that is, it happened before the split between species. And so that's when it happens. And so it's a very interesting and divisive thing to say in some crowds. And all I can say is that it's the observation. And one can live with it and have it and grapple with it however they would like.
Is that a strong immune addition, or is it?
I don't believe there's any directional selection there. That is, one is no better than the other. It's just simply being really diverse is a good thing. Having a lot of variety is a good thing. And so a lot of variety has been preserved at those locations.
Thank you.
Sure. So this down here is a screenshot of what one of the browsers might look like. So here is the depiction of the chromosome. And here is that narrowing neck indicating the centromere. And you've got the telomeres out on the end. This little red box then, as you're surfing through, is actually what's blown up down here in the little red box. And so you've got multiple levels in the genome browser where you can see all these different features. Here in this gold color are actually the genes that I showed in black earlier. Not the same genes, but the same information there would be on that track. And then all of the other layers of information that we're gathering now. Expected methylation states, if you're talking about epigenetics and the other physical structures that are involved, those are all layered on top. And there's a tremendous amount that's literally publicly available. And it's kind of fun to play around with, actually.
So this picture we're looking at, the purple and the blue, that's the banding of the chromosome, and then the gold is the genes.
You're exactly right. Yep, and this is a very fine scale banding, but yes, yep. So this is, so let me actually pull back, because there were a couple things points I wanted to make about the genome sequence itself. So it is not one human being, obviously, it's a haploid genome. And it was also not derived from a single sample. There were 13 anonymous donors in Buffalo, New York, that contributed their DNA that got put all over the world to the multiple locations that did the sequencing. The way they did this was very, very long chunks in yeast artificial chromosomes, and because they had long read lengths, that is many, many base pairs, they could overlap them pretty easily. And so what they did was build this scaffold that, if you remember when Craig Venter and Francis Collins were beating each other up on the cover of Time, Venter was using the scaffold that the government was producing the hard way to do something that I'm going to show you now. He was cheating. And it's still the way that we work today, where we have this scaffold. The reference genome is an expectation of what one should observe at a particular location, and it's the one we all agree on. But it doesn't mean that it's the best. It's the one that we all agree on. And in some ways, it doesn't matter which one, so long as we all agree. So there are mutations, what we would call clinical mutations, in the reference genome. And so that gets a little weird to report, because what is normal? There really isn't anything that way.
At the last conference, what is really normal?
It's a really interesting discussion that could go on for hours, quite literally. But I've heard clinical geneticists very upset that the reference genome should be the normal allele. And that conversation quickly devolves, because what's normal? So it's a reference, it's a scaffold, it's always being updated. There are different versions of it. It is, as I said, not a reading left to right, top to bottom of the entire set of base pairs. It's not the whole encyclopedia in that way. It's sort of cut and paste from World Book and Encyclopedia Britannica and what was the AOL version and Wikipedia. It's all these chopped up little pieces all crammed together, but it's the amalgam that we all agree on. And that's what makes it really powerful. So this is why it's powerful. So this is a technique called shotgun sequencing. So at the top here, what I'm showing is the expectation. That's the reference sequence. That's what we should see. And so you go in then and you take a sample from you or I or whoever, and you chop it up into little bits. Big enough that they're usually unique, but small enough to efficiently amplify, and that are small enough to actually use the mathematical algorithms that try to place them on the scaffold. So I'm blanking on the game show now, but it used to drop in and the letter was at Scrabble, where it used to like go brrrr, and then it would drop in the place where it was supposed to be. It's very similar about what we're doing here. We're taking the small chunks and we're trying to figure out where in this expected scaffold things fall. But the first step is to chop everything up into tiny little pieces, and then amplify. And so what you're seeing is that some of these pieces have overlapping information, right? So they all collapse back. If you were to take all of those things from the bottom up and sort of roll them up, they would collapse onto the reference. And so what you can see is that some of them are only sampled once. So this A over here occurs on only one of my reads. The one here in the middle occurs on many, many more. And so the way the reads align, the more that align, the more confident you are in your call, especially if you've got a half and half. So if you saw, okay, so let me feed forward a second. So this might be an alternative set of reads. So what I'm doing is coding polymorphisms. So this, we expect a T, but we see both T's and A's by the way it lines up. And so we infer that from mom came a T or an A, we don't know which at this point, without sequencing her, and from dad came the other. And so what you can end up doing then is calling these as SNPs, single nucleotide polymorphisms. You can also see now how read length makes a difference. If I had only one read and I saw a G, I don't have any information about what's on the other locus. If I have two and they're both Gs, I'm still not confident that there's not something else there. So the more reads you pile up, the more confident you are in your call. And this is a mathematical algorithm that's been developed. People differ in their algorithms. There are some that are optimized for certain regions of the genome because the composition, the expected way that things pile up is more tractable for some than others. So there's also, so the calling of a genome, the actual, I won't call it guessing, but the calling of the base pairs that are actually present is a process very often like this. There's a corresponding array technology that I'll tell you, and I'll pass these around now in case we don't get to it. So these are an alternative technology. They are microarrays. So there's one that's got a little bit bigger chip here and one that's got a little bit smaller. Adhered to those chips are pieces of the reference genome. They're cherry-picked pieces. They're signposts. They're places that we know are variable and fairly common variable, that is, 2 to 5% or more allele frequencies. So not the very, very rare stuff. So these are research tools. Well, so the DMET is a little bit different. The DMET does have some of the more rare ones because they're functionally relevant. Those are the ones that have effects on drug metabolism. And so no matter how rare they are, if we see them, we know what they mean. The larger chip here is more of a survey of the genome. Every couple of miles. Imagine driving from here to Los Angeles, and every 5 minutes you open your eyes. You would know that you were in the mountains. You would know when you hit the plains, but you're not going to know every weed on the side of the road all the way. But you nonetheless have a lot of information that you can use. So a similar technology in some ways, but very different in its chemistry. It targeted at actually known pieces of the reference genome. So feel free to take a look if you're interested.
Please. How is this used clinically? Is this a research tool or a clinical tool?
So the chips or the reference sequence?
The reference sequence, the shotgun sequencing that you're showing.
So when clinical labs are doing clinically certified, and there is a difference between research and clinical in the number of reads you're required, in some of the quality control metrics, the repeatability, there's a much higher standard when it's clinical, but it's done very often in the same way.
So they're looking for an abnormality in a gene site or loci. They're looking for, are they using that for a diagnostic tool or just to see how close you are to the reference?
So it depends. So when there is a known mutation that we know has an effect, so let's say you were doing carrier screening, where you wanted to know all the different inborn errors of metabolism that your child might end up with. We know when we see it, it's a simple architecture. When we see it, there could be a problem. And so in that sense, it's diagnostic. The presence of the mutation means you will have the trait, or it means that you should be managed in a clinically different way. And then there's the whole gamut moving out from there.
So how soon would you get that? How soon would you give it to a child? Or is this something that's done very early on? Or?
So it's very often done to end what's called a diagnostic odyssey. So this kind of whole genome sequencing really is not very useful clinically right now. There are all sorts of ethical issues, regulatory issues. It's a very, very complicated space right now. When something like this is justified might be when there just is no diagnosis. We have no idea what's going on with this kid.
I see.
And then you would look at anywhere and everywhere, because you've already looked at the places you know.
Okay.
You already have interrogated the things that you think are wrong, and you've come up empty.
Okay.
And so then you use some of these technologies to look a little larger.
Okay.
I'll also just briefly mention, because I know it was mentioned last time, exon sequencing. So what you do there is actually chop the genome up and pull down only the pieces that you know are associated with exons. So it's another step in the chemistry that separates the pieces that you're interested from the pieces that you're not, and then you use the same technology. but you have a filtered set that you've applied the same technology to. And so then when you build it back, you know it's going to build back to exons and not to just anywhere in the reference. So there's lots of different twists and turns on this kind of thing, but it's sort of all, it's philosophically similar, I'll put it that way. So this is my depiction of haplotype, of what we talked about earlier. Again, it's my soapbox, so I always try to throw this in. If you observed simply like an A and a T, a G and a C, and a G and an A at 3 separate locations, so I'm suggesting that this blue and red are alternatives, pick whichever letter you want, alternatives, alternatives. What you see very often are these calls, right? You don't really know if the capital A and the capital A and the capital A are all in the same place or not, because you're dealing with small pieces. If you read across the entire gene, you would know. But the first step in lots of these sequencing technologies is to cut down to 100 to 200 base pairs. Some of them go to 300 to 400 base pairs. There are some technologies that can do thousands. And so then if the mutations are within that read length and you've got enough overlap, you can be pretty confident as to which ones are traveling together. But when you get down to some of these shotgun type technologies that rely on shorter and shorter read lengths, you lose that information. Those chips that you're looking at also lose this information. They're guideposts. They're meant to be physically disparate. And so when you get those answers, it can be difficult. And there are some assumptions you can build in to make the call, to make your best guess. But there are some current limitations, and we don't always know. Okay, so this is my transition slide from genetics to genomics. And so what I want to point out is that the use of genetics in the clinic is not new. It's not new at all. So one of these very simple, thought to be Mendelian type characteristics, PKU, phenylketonuria, has been screened since the 60s with Guthrie cards. So it's using the enzyme activity or some other features of the protein to diagnose the underlying mutation, but it's still pretty much genetics, right? And then all sorts of other things have been introduced since. Molecular genetics in the 70s and 80s gave us lots of these classic examples of a Mendelian character that ended up being useful clinically. Because when you see them, you end up with a trait. I'll just throw out that sometimes your sample matters. Okay, so someone has a physical characteristic, let's say it's an enzyme deficiency. I've seen it before that people with that enzyme deficiency have the mutation. So you then go into that individual, you either sequence or you use an array or you do whatever, you confirm that they have that mutation. You've now reestablished and reemphasized the link between the mutation and the trait. But what have you done? Where have you looked? Only in the people with the physical trait, with the actual phenotype. When it turns out that when you go out and sample more broadly in the general public, it happens where someone has that marker and does not show the trait. And so even some of the classically Mendelian characteristics are things that really need to be careful. And so then precision medicine becomes this interplay between one or the other. Personalized or precision, I should note too, you can use pretty much independently. With the recent State of the Union, everyone's saying precision now. We picked personalized early on, but it doesn't really matter. It gets at the same idea. And it's a big idea too, which we can also talk about. But it ends up being this sort of push-pull where you need complementary types of information. My view, and I won't speak for anyone else, but my view of genomics' role in medicine is not at all as a panacea. It's not at all a silver bullet, be-all and end-all that all we got to do is sequence everybody, and we'll be fine. It's added information that gives the doctor-patient interaction more meaning and more framework and more possibilities. It's really a decision support tool, one that helps a doctor and a patient decide on a course of treatment in the same way that some of the other features do that they measure that are non-genetic. And so the idea that if we just sequence everybody we'll be fine is just not bearing out, especially with all the gray area that we've already covered.
And does that information really help only if there's a severity of the patient compared to what you expressed, that a person may have something but is not visible, but they're carrying it. So on one side, the person dealing with preventive health don't really have enough information to help them be preventive. And on the other side, the doctor can't provide information because there's no actual physical manifestation of the variation.
It's all case by case. It's really, it's all a continuum. And there are some cases, like I said, where it's clear that you have benefit. There are some cases where it really is sort of if you're really just going to throw a dart at the dartboard, you might want to throw the dart with a little bit of an intention. And more information can give you a little bit more of an intention. And so one of the pieces of information that might be useful in that is genetics.
And I was just thinking going from, I mean, in some cases too, I mean, that information, unless there's a specific, there's probably only some specific medicines, it doesn't necessarily matter. It just gives you more information to say what your diagnosis is, not that we can help you. You know, right?
And so again, my work that I really enjoy is on conditionally dependent features, things that it's not they're necessary but perhaps not sufficient. That is, when you see them, it doesn't mean that you will definitely have it. There's a context involved. Everything has a context dependency. So gene-gene interactions are a form of context dependency. Gene-environment interactions are a form of context dependency. And there's some really tricky things that happen when you get interactions like that, where you need more than one thing to be true to see the outcome that you expect. My thesis chair used a term that's never caught on for obvious reasons, but I think it's instructive. He called it the confoundment of frequency and causation. That is, the rare thing looks causal when you've got an interaction system. Give you an example. PKU is a widely considered genetic disease. If you have the mutation, you end up with you have the propensity to end up with this. Why? Because you can't metabolize phenylalanine. Phenylalanine is ubiquitous in most diets, so it's the common thing. It's everybody has that dietary component unless they're very careful about it. So then, when you have the mutation in an environment where you're taking in the nutrient, you're going to end up with the trait. But it's still a gene by environment interaction. It doesn't necessarily mean that it's a purely genetic trait. Flip it around. Scurvy is widely thought of as an environmental disease, right? You get on a ship, you don't have any vitamin C, you go out to sea for a couple of months, your teeth fall out. This is something that was a very obviously environmental thing, because that didn't happen when the sailors were back in port. But it turns out that we are all homozygous for a non-functional enzyme that makes our own vitamin C. Most organisms on the face of the earth can make their own. There are some very odd exceptions. A lot of bats, perhaps because they tend to be frugivores and they're eating a lot of fruit, and so then the mutation arose and it just got fixed. Guinea pigs, if you've ever had a guinea pig, you have to provision with vitamin C because they can't make their own. And then I think it's called the Haplotypes. It's this whole clade of monkeys and apes and humans. So it's the clade that includes humans. At the base of that tree, we became homozygous for a non-functional version. So again, the common thing is to not have the genetic part. So the rare thing then is the environment, where there's almost always enough vitamin C around. So it's this very interesting thing where people talk about nature versus nurture. It's clearly not to me. To my mind, you can use anything else that you want, nature via nurture, if you prefer. But the interactions between genes and environment are a fundamental feature of traits. When you have DNA in a test tube, it does not have diabetes. It does not have a blood pressure. And so there is this context dependency of it being in a viable zygote that goes through the developmental process, that then has an age and then has all this other stuff attached.
My daughter has scoliosis, and we just went to, her last, seems to be she's been sort of excused. And, the doctor said to us that there's a strong genetic component. And when I read up on that, that is not necessarily what I thought, because neither my husband nor I have, so when he said strong, the words he chose to use to say that, I thought, I felt blown away, because in the research it says idiopathic. So there's definitely a genetic component to it. Yep. But when you say strong and dominant, I don't have it, my husband doesn't have it. I'm saying, that we have the manifestation of the disease. Sure. Obviously, she got the genetics from somewhere, but then what happened during development, and all that, that's those kinds of things, or what?
So another way to think about that is a tendency to run in families. It doesn't mean that everybody has it, but it tends to run in one's family. And then it's really unclear the actual source of the function.
That's what I was kind of thinking of.
Now, just wanted to ask 2 questions. Muscular dystrophy and bipolar disorder. My daughter-in-law has this she somehow measured some genetic screening that put her over into the middle range of muscular dystrophy. I'm just wondering if you could comment about that. I don't understand.
It's not really my area.
Oh, okay.
I wouldn't feel comfortable saying a whole lot because it's really not my expertise.
There's some sort of genetic determination about how bad your muscular dystrophy is.
I'm sure that there are some indicators of mutations that are associated with a higher penetrance, that is either a more sure manifestation of the disease or some sort of progression trait.
Have they identified one for bipolar disorder?
So the genetics of the neuropsych disorders are becoming very interesting. There are lots of hits when one looks genome-wide for these things, but it looks more like an obesity type trait. Lots of different loci of very small effect. At least that's the preliminary idea now, that there really is no single mutation that's likely to cause a lot of these neuropsychiatric conditions. That it takes a multiple combination. And then it's a matter of spinning the dial, you see where you end up, and then you've got your environment and everything else layered on top of that.
The thought that comes to me now is the question about interpretation as you deal with this. Is the doctor, the practitioner, the one that's really doing the interpretation, and therefore, or is it another information system that is doing the interpretation that the physician is utilizing in order to help in the diagnosis?
It remains to be seen. So it depends on who you ask as well, with the regulatory environment and how exactly this is going to be brought around. We do have a set of professionals that are trained in this. They're called genetic counselors, and they're often very lost in this conversation. And it's always been baffling to me because we do have a trained, group of professionals that can do this. But there's a little bit of arm wrestling going on between physicians and genetic counselors, pathologists who have been very big in the cancer genetics area, that maybe aren't the right ones perhaps to have be looking at some of these other more complicated situations. It's unclear if it should be FDA or who exactly is going to be doing this oversight, and it's kind of a hot potato. So I'm mentioning a more utopian situation where information ends up in the hands of an expert in a dialogue with their patients.
Yeah, y'all hurt me over here.
So it is a little more complicated. Yeah. And of course, I like to throw up examples too. This is what we're dealing with, things like these pathways. And so you can start to talk about architectures too, where if you're just missing the receptor, it doesn't matter what happens downstream. Yeah. If it just isn't there, that's an example of a choke point that you really might actually be clinically useful. But something down here that's playing around with multiple redundancies, and there's lots of different ways to get to the same answer, a mutation there really might not be as useful. And it depends. So I'm conscious of time. We're getting a little close. Let me just quickly go through this because I think this is also sort of useful. It brings home part of our conversation. So clinical diagnoses are really all or nothing events. And so we talked about cases where you've got a diagnostic odyssey where you don't have a diagnosis. You need to come up with them. So that's one kind of aspect of this. But when you think about risk of disease for most of the things that we're talking about and all these different features of the genetic architecture, and I include environmental interactions in the genetic architecture, it really becomes this continuous variable and not a dichotomous trait. And so then the challenge is to see where in the continuum you actually fall. And so I like to use this. This is a frequency diagram where on the x-axis here is the trait of interest. It happens to be height. And on the y-axis here, you've got the frequency of the observation. So there are 5 people that are short, that are 5 feet or between 5 and 5'1". And there are, 3 here that are 6'5" or above. And so what you can see is a normal distribution. If you look more closely, you can see 2 normal distributions. So see those in dark colored sweaters. And if you collapse down the ones in white, sort of like Connect 4, you get another distribution. That's a dichotomous variable. That's the women and the men. And they each within them have their own means and their own distribution. And so this is really what we're dealing with complex trait variation. And so the trick is that we've got this x-axis now is this abstract continuous variable of risk that might not be easy to put your finger on. It's not the presence of one thing versus the other. But hopefully you can end up finding the right combinations of things that do put you in a bucket that includes others with high risk. And so you can imagine with 1 locus and 2 alleles, that's what you would expect. When you add more loci, it starts to fill out. And this is how they actually reconcile the Mendelian traits with continuous traits. Early on, there was a pretty fierce debate about whether there were different kinds of variation. what sort of function, what sort of mechanism is giving rise to these. They're totally different types of traits. But this synthesis actually brought things together in the early 20th century that no, this is one way to explain it, and yeah, it does a heck of a good job of explaining it. And so this became, this plus the environmental variables that sort of smooth the curve, and all the other mechanisms that you can imagine. So then the idea is who's in this N, who's at high risk? And there could be multiple combinations of who's at high risk. And it's unclear, it doesn't have to be only the ones that have capital A. It could be spread throughout the Punnett square. There really are no rules there. I don't think I have any expectations, unless you know something about the architecture, something about the signaling cascade, something about the way that the body, the physiology actually works. I'm not sure I have an expectation. In some cases you will. In most cases you likely won't without more research. So okay, sorry, I got one more thing. Do I have one more story? Because I also do love this about genomics too. So I use this in many of my lectures to make a point about ancestry and about what your genomics tell you about where you're from. So this is a cartoon of my own. My mom is of Central European extraction. Her parents and her parents' parents and her parents' parents' parents were all from Poland. My dad's family is English and Irish. And so I've coded this with sort of green in honor of Ireland, sort of this red, I don't really know why I picked red for England, and this sort of bluish for Central Europe. And so what happens is my great-grandparents obviously had my grandparents, and recombination events took pieces of their chromosomes and swapped them over. So the pieces they got from mom and dad get shuffled. And so this happens with each one, and you move across, right? So my grandparents then moved to Grand Rapids, Michigan. So they moved from these physical locations to a completely new physical location. So, and then had my parents. So my question that I love to ask is, okay, so where is this piece from? It's from Grand Rapids. We sampled it in Grand Rapids. But you can actually, if you're able to go and sample with these other populations, you can see that it's actually this piece of this lime green color. But what about the piece right next door? Where did it come from?
England.
Yeah, England, right? So each locus in the genome has its own genealogy. It has its own history. And we are all an amalgamation of a whole series of genealogies. It is true that there are some things called private mutations, where you only see that mutation, at least so far as we've sampled, in one physical place in the world. We've never seen it anywhere else. And so if we see it in a person, we can be reasonably confident that they shared relatives with people from there. But it doesn't mean that their whole genome is from there. So I also like to say I've now continued the tradition, and I live and work in Camden. So it ends up being a pretty tricky story, because there are parts of me that have a high frequency in Central Europe, and parts of me that don't. And we're all that way. And so it comes down to, we're really all from everywhere. This is sort of the sameness argument. Very often geneticists and researchers love to say how different we are, because those are the fun stories. And those are the things that get you somewhere, get you funding, for example. But really, we're kind of all from everywhere, and we share an awful lot more than we differ at. I'll stop there.
Thank you, Jeff. That was great.
Thanks so much.
Last questions?
Of course, I have another question. Okay, so I teach genetics because I'm a biology teacher, and now every time I come to these things, I'm thinking, I'm giving these guys, information like it's the end-all be-all, and then here's this whole genomics piece that I certainly don't have the capabilities to teach. I don't have a textbook that supports it. I don't really, without going back to school, feel I have the, the ability to teach that. So how do you reconcile in a classroom where you are teaching genetics, the value of that, with all this other stuff that's really, even like last session, I think the person, maybe it was 2 sessions ago, said that our phenotypic expression is really our DNA, our genotype, only really about 10% of our genotype really codes for our phenotypic expression. So there's all that other stuff. So, I think to myself, how do you reconcile that when you're trying to explain, it's easy to teach genetics, but I teach this, dominant, recessive, mom, dad, this, that, and the other, and it, in a sense, in light of genomics, seems like trivial.
So let me first say that it's tricky, and it's one of the reasons I have such immense respect for you all. That it is sort of difficult to navigate this. One of the things that helps is to think about it in terms of a historical progression.
Yeah,
So I actually really love to show that the stuff that we have as our examples that we've brought down really were the gold standard, and they were useful. They got us to where we are now. Even if, and they're still true. It's not that they're not true. They actually are building blocks. They're the raw materials that can help you mix and match and put concepts together to find something new in the world. So the expectation that one can teach a subject and actually cover it is the problem. And I know that expectation comes from way beyond the classroom. But it's nonetheless incredibly useful to share with students the same building blocks that scientists were actually having vicious debates about 100 years ago.
I mean, could the comparative be that, they used, when you were saying that the 10% use, I immediately think of the brain. We were always taught that the brain, only 10% of it was actually, and now we know it's not. So,
Yeah.
But didn't stop us from teaching what we thought the brain anatomy was at that time.
Simple does not mean useless. Simple examples actually are very powerful, if only to show you what things are not. So that's sort of the Popperian paradigm, right? So it's the A, not A. It's what we're familiar with, or it's what we're not familiar with. And so it's building the backlog of the things that we're familiar with in order to define the ever decreasing subset of what we're not. So there's always going to be some of that. That's the fun part of this. And it doesn't mean that what we do know or what we think we know now isn't useful. And doesn't mean that some subset of it isn't useful in the clinic now. It's just not that everything. It's just that not everything is going to be that way. It depends on all sorts of factors, and that's both an exciting situation and a challenge. Because theoretically, the students are those that will be able to go out and contribute in a very real way. The closing of the book and the pretending as if we know everything actually is so stunting. And so, sorry, go ahead.
I just had a question. When you have genomics and personalized medicine, so are there clinical trials now structured where they have the reference coding for that chromosome? Okay, mine might be different from hers. We're giving the same dose of medicine. We're looking at the efficacy of that drug. Is that what you mean by the personalized, genomics of personalized medicine?
Yes. It comes down to getting you the best treatment for you that we can. And there's all sorts of features that come into play about you as a person, and your genetic code is one of them. Some of that information can actually get you to a better place. And so personalized is also a little bit of a misnomer when you think of it that way. Because if we have only one observation, if you have a mutation that no one else in the entire world has, that's never going to be clinically useful because we can't demonstrate that it actually did anything. So personalized is really getting to I use the term bucket, right? So it's a grouping of patients that are alike enough that they can receive a tailored
So it's not
treatment
personalized,
trajectory.
it's more like the group with a cohort of people with certain traits, and that drug works on that group versus another cohort.
That's right.
Something else.
Not
personalized.
So that still is personalized because right now, think of the difference, right? So right now, you're just getting everything. You're just getting something random. Well, this is what we usually give people. That's very impersonal. So no matter whether it's you individually or you as a class of patient, it's still more personalized to you because you're a member of a group to which it can be targeted.
Right.
And so also to briefly answer your question, the drug companies are kind of figuring this out, and so they are structuring lots of sequencing into some of their trials. But really a lot of this has happened by accident, where individuals are given a standard treatment for chemotherapy. Some of them die. Some of them go into remission. And then in retrospect, they go back and are able to pick out the difference between those two groups. So there's a lot of retrospective work that's been done. That's the basis of most of the stuff that's pretty well understood. But now they're sort of coming at it from both ends now, where we're doing more prospective type.
Are they able to do any corrections such as sickle cell and chromosome 6 or anything like that? Are they able to manipulate any of those genes to fix that yet?
So there's a lot of work in this area, and it's really tricky. And so nothing that I'm aware of is actually clinically useful. There have been a couple of studies that looked promising. There's certainly some technologies that will allow you to do that, but there are perhaps unintended consequences. It also is that you have to get into every cell. So we talked about us being an amalgam of different genomes, right? You have to get into every cell type or maybe just the cell types that the disease is actually a problem in. So in your example, just the blood cells. And so that seems like a fairly tractable place where we might be able to fix just the blood cells if we can find a way to target just them. And then we don't have to worry about the sickle cell and any other tissues. So it's promising, but it's not yet ready for prime time. There are lots of challenges there.
I was just wondering if there's anything that, any combinations or genomic combinations that code for higher IQs in any of the areas, now that take the different intelligences.
So there's some really interesting work. Actually, there's a great example that I love to show about IQ. So there is a very, very strong what's called heritability of IQ. And so what that means is that when you think about that distribution, wherever your parents fell on that distribution, you tend to fall in the same general region, high or low. What tends to be lost in this is this environmental component. If you take that same distribution and you slide it way to the right because of all sorts of educational advantages and all sorts of socioeconomic issues, you can be at the left end of the distribution and have a much higher IQ than most of the people in the world. And so it both has a genetic component in its sort of underlying distribution, but it's not meaningful for predicting a specific value of your IQ.
Right.
At least now. There's many other variables that help do that. And so that's in the same way as some of these obesity and coronary artery disease type loci. Yes, it's true, it's genetic, but it's not. That's only one piece of information.
I want to take you out of what you're presenting and in line of students, because I'm thinking K to 5, right? And the one piece that you meant about the genomic technology and pictures, because I'm thinking, how do you introduce this at an early age to students so they have a better understanding as they develop by the time they get to an earlier age, and so in trying to create a science program within the school. So have you ever had an inclination after going through all this study and doing all this work of, if I was able to go into a K to 5, K to 6 school, how would I organize it?
Sure. So I use the analogy of the telescope. So one can imagine you're looking up at the stars with just your eyes, right? So now we have a new tool that's able to see in a way that we couldn't see before. And then now we've got the Hubble Space Telescope, or we've got a radio telescope. We've got all these different technological innovations that take something that you already have an experience of, and the technology then enables a different frame of reference. New things become apparent because you're able to look in a different way. So I think any technology or any sort of process that is sort of expanding like that is the same as what's happening here. We've developed some tools and techniques that have allowed us to look at the genome in a way that we could not look at it 30 years ago. And it's the same sort of consequences that we had when we had the telescope, and then we had bigger and bigger telescopes, and then people got yelled at pretty good for saying that certain things were true because they were looking with a different technology.
So I interpret that, and I'm interpreting that, and I'll stop on that because that's one reason why I came to these sessions. But for that K, maybe a certain level of pictures, a certain level of technologies, a level of understanding just shapes, and then going into so that by the time they get to where they have to by science, that it's already a progression to do that. Okay.
I think that's great.
I got to sell it, but okay.
So not just magnification. So I was suggesting that magnification was one of them, but resolution too. So you might be able to see a long ways away, but it's fuzzy, very, very fuzzy. So once you zoom in, you still have to increase your resolution. So there's sort of multiple parameters there where you're actually sort of circling in on the truth because of these different ways of accessing it.
You got to make it fun.
Yeah, it is a challenge, isn't it? Try making statistical genomics interesting. It's tough.
I have another question, just related to when you said that there is no, we haven't seen in the reference piece, no real norm, no real one norm. So that, as soon as you said that, was kind of like, this is where the evolutionary piece, there's no norm because there's not, there is nothing like that. So when you are studying, when you're doing all of this, do you see certain genes or certain areas that have actually seen evolve over some sort of time? Because kids always, when I have to talk about evolution, well, then that's a whole other, we don't see any of that. Or you're, telling me that apes are going to turn into humans, and I said, well, it doesn't really work like that. And I always use bacteria as the example of there's evolution. Yes, there is evolution today. The reason we can't see, we have very long life cycles where bacteria don't. So I just use that as an example of survival of the fittest. This is what staph was supposed to do. It was supposed to become resistant to it manufactures ways of. Well, anyway, I don't. But that's my only example. I don't really have examples to talk about in a human.
So the trickiest part of that is that fitness is not performance. Fitness is a trait that is how many kids you leave behind. It's how much of your genetics is represented in the next generation.
Right.
That's high fitness, not someone who is a prime specimen of humanity.
Absolutely.
And so then you start to look for these examples where, very sadly in human history, early mortality, early childhood mortality are places where if individuals do survive more often than not through childhood, there's a better chance for the individuals that have that stretch to contribute to the next generation because they've made it past childhood.
Right.
So we already talked about the sickle cell example, where in an environment where there's malaria, it actually is very beneficial to get past that childhood age. In an environment that does not include malaria, it's not a very good thing to have. One other example that you can point out is lactose intolerance. Is a classic example from pastoralist populations that domesticated cattle. So now you've got this situation where if your prime food source is something that you can't digest, that's not likely to be a good thing for you. And so it's also persistence, right? Because we're all lactose tolerant to start out with, but we shut it off. There are some populations that leave it on. And so that mutation that left the lactose tolerance on, the vast majority of the world is actually lactose intolerant. It's all Europe, small places in Africa and Northern Europe. And so it's
that's a really good example. And I just did something with my students where we did that hypertension study of African blacks versus American blacks and the sodium retention that perhaps, and of course there's probably as many documents that say, well, that's a bunch of bunk but I did pick one that said, just to look at the pros and cons of salt retention. And it could certainly be a pro if you have to cross an ocean and have deplorable conditions, that it was a survival kind of thing. But then on the con, if that's actually true, then could lead to a hypertensive state.
So there's also an example that's called the thrifty phenotype hypothesis. So in times of famine, the ability to retain nutrients and be really thrifty with your metabolism means you're probably going to survive the famine. So in the presence of famine, you assemble all of these alleles in populations that are experiencing it at regular periods. The famine washes through, and only those kids that have the combinations to be able to make it through are the ones that end up having kids of their own. In an environment where famines don't occur and you have a surplus of food, they cause obesity. Because now you don't have those regular periods of nutrition starvation, and the same sort of, so this is that gene-by-environment interaction piece. And so selection is also a very, very difficult thing because it's time and space dependent. And so the selection regimes that are likely to be manifest in humans, in my opinion, are those that target early life, infectious disease, big time. I can see the Black Death. I can see variola major. What am I thinking?
Chickenpox?
What's that?
No, you said variola major.
It's variola. It's smallpox. Smallpox was nasty. It killed a lot of kids. And so I can see that being a selection pressure, and things that then allow you to beat the smallpox virus would be things that get carried through and then passed on to your own children.
That's a very good example.
But things
about Ebola a lot too. I said not everybody dies from Ebola, but not everybody.
So, in a social species, why do so many of us have terrible vision? Because by the time our vision really degrades to a point where we really can't function, we're post-reproductive. We've already had our kids, or we can help each other out. And it doesn't matter if you can't see because your kids can. And so then there's this sort of this piling up of, so you never select that out. Late onset diseases are likely not the product of evolution at all.
Yeah.
something like Alzheimer's,
Yeah.
Parkinson's, how would that ever possibly have been under
Right.
selection?
Right.
It's
Right.
way post-reproductive in
Right.
most cases, especially when the average human generation time is on the order of like 15 to 20 years
Yeah.
over the course of human history. You're done reproducing by,
Yeah.
20, 30 years old.
Good examples, thank you, that's something I can.
Sure.
Yeah. When are you speaking again?
I talk all the time, usually to myself.
Welcome to the club.
Hope this was helpful. I had a great time. Thanks so
Thank you
much. It's
very
such a
much.
privilege.